Welcome to section 6.2, the cell cycle. In this section, we'll connect the big picture, why cells divide at all, to the details of how they move through interphase, mitosis, and cytokinesis. We'll also look at the resting state called G0 and the checkpoints that keep division accurate. By the end of this section, you should be able to describe the three stages of interphase, G1, S, and G2, discuss how chromosomes behave during mitosis and how the cytoplasm divides during cytokinesis, define the quiescent G0 phase, and explain the three internal control checkpoints at the end of G1, at the G2 to M transition, and during metaphase. The cell cycle is the ordered set of stages a cell follows to grow and produce two daughter cells. Most of a cell's time is spent in interphase, preparing for division. When conditions are right, the cell enters the mitotic phase to segregate the nucleus, and cytokinesis then splits the cytoplasm. The key idea here is that cell division is controlled and cyclical, not random. Proper timing ensures that each daughter cell receives a complete genome. Interphase has three stages: G1, S, and G2. In G1, the cell grows, performs normal metabolism, and builds up energy and molecular resources for DNA replication. During S phase, the cell synthesizes a complete copy of its DNA. Each chromosome is replicated to form two sister chromatids. In G2, the cell continues to grow and synthesizes proteins needed for mitosis, double-checking that DNA replication is complete and ready for accurate chromosome segregation. Mitosis divides one nucleus into two genetically identical nuclei. Briefly, in prophase, chromatin condenses into visible chromosomes and a spindle apparatus forms. The nuclear envelope breaks down. In metaphase, chromosomes align at the cell's equator, each chromatid attached to opposite spindle poles. In anaphase, sister chromatids separate and move toward the poles. And in telophase, nuclei reform and chromosomes decondense, setting the stage for cytokinesis. Cytokinesis physically separates the cytoplasm. In animal cells, a contractile ring under the plasma membrane tightens to form a cleavage furrow, pinching one cell into two. In plant cells, vesicles from the Golgi apparatus coalesce at the center to build a cell plate. Membranes fuse and cellulose is deposited to form a new cell wall, completing the partition. The mechanism differs, but the outcome is still two distinct daughter cells. G0 is a quiescent state where cells exit the cycle and stop dividing. Some cells rarely or never enter G0 because they divide continuously. Examples of continuously dividing cells include epithelial cells, hematopoietic stem cells, spermatogonia, and many cancer cells. Other cells enter G0 temporarily, such as liver cells, white blood cells, and fibroblasts, which can re-enter the cycle when needed. Some cells enter G0 more or less permanently, including neurons, cardiac muscle cells, skeletal muscle cells, and red blood cells. Cell cycle checkpoints are quality control gates. The G1 checkpoint asks, is the cell large enough? Does it have sufficient energy reserves? Is the DNA undamaged? The G2 checkpoint verifies that all chromosomes have been fully replicated and checks for DNA damage before mitosis begins. The M checkpoint ensures each duplicated chromosome is properly attached to the spindle and aligned at metaphase, preventing missegregation. These controls protect genome integrity. Interphase builds the capacity to divide, growing, replicating DNA, and preparing machinery. Mitosis sorts and parcels identical copies of the genome into two nuclei, and cytokinesis completes the split into two daughter cells. G0 provides flexibility, allowing cells to pause division for specialization or maintenance. Throughout, checkpoints safeguard fidelity so that daughter cells inherit complete, accurate genetic information.